Diet is crucial to understanding ecology and, by extension, evolution. An ecological position of an animal tells you exactly where it was in its ecosystem and what the disappearance or evolution of its food meant for its own evolution. It is very difficult to understand the ecology of a given organism when all you have of them are fossils, but sometimes you get a peek into what the hell they were doing with themselves if you understand how chemistry works. Such an understanding of chemistry was just recently used to extrapolate on what is known of the ecology of the largest shark to ever live, Megalodon. And it shows that Megalodon and the great white shark are more similar than previously thought, and in a way no one had imagined. One of the biggest problems with paleontology is trying to reconfigure, discombobulate, recombobulate, and just plumb figure out the ecological niches and trophic levels of extinct creatures. In other words, their ecology. This even includes apex and mesopredators as well known as sharks. Sharks have been around longer than the dinosaurs, crocodilians, and even trees. But even with such an old lineage, their ecology can only be inferred from fossil stomach contents. Sharks have a horrible fossil record too, thanks to having a completely organic skeleton of rubbery cartilage. So full skeletons are impossible except under very rare and special conditions, and even just the skull and spinal column are rare too. Teeth are the most common parts preserved as they are the only part of their skeleton that contains organically secreted inorganic compounds like dentine. When it comes to ecology, diet plays an essential role in the evolution and extinction of a species. Interactions between trophic levels in an ecosystem can stimulate competition amongst species, while the loss of a prey species may have detrimental effects to a species' survival and result in extinction. Just for a refresher, the trophic level of an organism is the position it occupies in a food web. A food chain is a succession of organisms that eat other organisms and may, in turn, be eaten themselves. The trophic level of an organism is the number of steps it is from the start of the chain. Anyway, figuring the precise nature of the diet and trophic level of any given extinct organism is a huge pain in the ass, and many times it is entirely impossible. The impossible part is usually when you have a completely unique example of an entire group and none of the teeth or skull bones were preserved. For your reassurance, this complete impossibility is also quite rare. Figuring out the precise nature of the diet or ecology of an extinct organism is hard and complex, and often relies on inferences one can make from anatomy or evidence left behind, around, or on the organism that is not the organism itself. Fossil gut contents, bite marks, or remains in fossil poop. These data bits are rare and preserve only under rare circumstances, so they don't really tell you a lot about a lot of different critters. On top of that, even when they do occur, they don't tell you about the diet of the entire species over a period of time, nor does it tell you where it was in the ecosystem, its trophic level. It just tells you what it had ingested or fought with before death. Ever hear of zinc? Them zinc tablets you may have taken when you had a cold? Yeah, well, those aren't pure zinc, shocker, because zinc is a metal element, so that would make it a hard pill to swallow. Fun fact, zinc is the 24th most abundant element in Earth's crust, and is an essential vitamin and mineral for all living organisms, part of this balanced breakfast. The element part plays a crucial role in various boring biological processes. like living, or more seriously, like bioapatite mineralization. So the hardening of the skeleton, zinc that is contained within the humid, seething walls of critter flesh, mainly comes from the animal's diet. In the breast-having, milk-squirting, hair-covered, oil-oozing mammals, isotope ratios of zinc are actually incredibly useful for getting a general idea of the trophic level that mammal occupies. Who would have thought? Well, mammalogists, I'd imagine. Unfortunately, studies on the various isotope values of zinc in marine mammal carcasses was only ever performed on marine mammal carcasses found in archaeological context, rather than freshly living ones. Regardless of that comparative hiccup, every single other study that links zinc isotope values to trophic levels show zinc values decrease with increasing trophic level. I guess that kinda makes some sense, right? As you go up the trophic levels, there is less bulk energy going from organism to organism. Plants have the most since they get it directly from the sun. Herbivores get less of it, and the carnivores that eat them get the least. Zinc isn't energy, but considering the successive decreasing relationship exists as you go up the trophic levels, I'll bet that plants contain the most zinc, and carnivores the least. The trophic dietary zinc isotope thing is used by most animals to help them produce the organically secreted inorganic parts of their body, namely bones and teeth. It is especially useful in the hardest enamel or enameloid teeth, which is incredibly resistant to change. It would make sense that organisms would need ultra-hard teeth made of material that does not deteriorate for their entire lives. And then it would also make sense that whatever that material is, in this case bioapatite or hydroxyapatite, would also survive just about every single fossilization process intact. That is why you find isolated teeth of all sorts of animals all over the place so easily. All that is well and good, but there are no known zinc isotope values for the teeth of anything that is not a mammal. That makes it difficult to understand the ecology of everything else. On top of that, no data exists for fossil teeth older than the late Pleistocene, which then makes it hard to understand the ecology of mammals and non-mammals from before that time too. In a brand new paper by a team of 17 researchers led by Jeremy McCormack, the zinc isotope values are examined and recorded from various modern sharks and fish from different locations. The team recorded the numbers from different kinds of tissues too, the enameloid and dentine of the teeth, the gill rakers, and the bone. They included about 20 living species into the study, which also includes species kept and fed in an artificial environment as a control. Then, the team investigated the deep time preservation potential of the zinc isotope value to trophic level proxy by including samples of different fossil tissues from various fossil species, including Megalodon, which I might add is a common name. The animal's real scientific designation is largely agreed upon as Otodus megalodon. Otodus megalodon is the largest and last species of the megatooth shark lineage, which began during the Paleoecene, only seeing its end at the end of the Pliocene. That just so happens to make it one of the largest carnivores to ever live too, though definitely not if you define eating krill as active predation. Everyone wants to know how something like Megalodon could go extinct. Diet and competition for resources with other critters have been oft discussed as possible drivers of extinction and evolution for the shark, but its relatively abrupt appearance and disappearance in the fossil record does leave a big gaping mystery hole of glory right there for the scientific taking. In the new study, the team used zinc isotopes to compare the dietary ecology of two species within the Otodus genus from early and late in the evolution of the genus to get an idea of how its ecology changed over time. Those two species, of course, being Otodus chuatensis, the older species, and Otodus megalodon, the younger species. This method was also used to compare these ecologies to the ecology of the great white, a species which has been around since the Pliocene. First things first, the team found that zinc isotopes are a promising indicator of trophic levels for both sharks and fish in general, in a similar fashion to how it is in terrestrial and marine mammals. It's probably relatively safe to speculate that this sort of indicator may therefore be present in reptiles as well. Wonder if there is a way to do something similar with toothless animals. The team analyzed the zinc isotope values for living sharks spanning captive/aquarium and wild individuals from various localities, and found close correspondence with their respective trophic level. Large apex predatory sharks like the great white Carcharodon carcharias, the short-fin mako Isurus oxyrhynchus, and the salmon shark Lamna ditropis have significantly more negative zinc values than lower trophic level fish and the plankton-feeding basking shark Cetorinus maximus. In particular, the short-fin mako and great white sharks, both of which are apex predators, have much lower zinc values than in any previously recorded living vertebrate species. These low zinc values are likely due to the larger number of trophic levels in the marine ecosystem than in terrestrial food webs, and maybe even differences between marine and terrestrial zinc isotope baselines. The team found that total enameloid zinc values vary by up to 1.26% among the living species that they analyzed from different oceanographic areas. There is also large variability in enameloid zinc values among living sharks within the same region. Just some fish food for thought, there is a 0.88% difference between mean values of great white and bull sharks from KwaZulu-Natal, South Africa. In contrast, enameloid zinc from the same species among great whites and dusky sharks demonstrates a relatively low level of variability within isotopic signatures, regardless of geographic location. When the team analyzed fossil shark teeth, they found similar levels of zinc isotopes to the living shark teeth. This suggests that very little affects the zinc component of teeth through the fossilization process. In other words, becoming a fossil doesn't create a bias or mess with the data. The team analyzed fossil shark teeth from Germany, Malta, Japan, North Carolina, and Florida, spanning the early Miocene 20.4 to 16 million years ago through the Miocene-Pliocene transition about 5.3 million years ago, and the early Pliocene about 5.3 to 3.6 million years ago. So, what do the zinc isotope numbers say about the fossil sharks? The team found that there were some species that did not change much in their ecology over time, with some modern species retaining similar zinc isotope values as their extinct relatives, while others differed more dramatically. For example, most shark species that have a slender, grasping tooth shape retained the same zinc values in both fossil teeth and modern teeth. The zinc isotope values found in the tiger shark lineage showed no differences between fossil and modern forms, indicating they have remained relatively stable in their ecology since at least the Miocene epoch. The earliest fossil tooth forms of tiger sharks are smaller on average than modern tiger sharks, but even here, the zinc remained at the same levels, suggesting a change in size did not change their general ecology. The living and extinct forms of the Carcharinus genus were found to always have different zinc values than all other sharks they live with, and this roughly remained the same over time. They also retained the same tooth shape and same amounts of zinc overall, suggesting they may have been living in shallow water and coastal areas, feeding on floating and swimming prey for most of their evolutionary history. Two species of otodus teeth were analyzed, Otodus chubutensis and Otodus megalodon. Analysis of zinc values in both species' teeth found that both species were apex predators that were a part of a very high trophic level. In every single early Miocene assemblage, the Otodus chubutensis teeth had the lowest zinc isotope values of any organism also found in the assemblage. Otodus chubutensis zinc values were found to be similar to those of the living great white, Carcharodon carcharias. What was a tad surprising was that the zinc values for great whites were different between the living and extinct forms. This difference indicates a dietary shift in the great white lineage. Now, quickly swinging our attention back to the Meg, Otodus Megalodon zinc values were rather different from the average in the population near Japan from the early Pliocene. They had much lower zinc values than Atlantic Megalodon populations sampled from Florida and North Carolina. A possible explanation for these differences may have something to do with prey preferences, or perhaps it was just baseline variation, aka they were just like that due to ever so slight genetic differences. The research team pointed out that they could not also rule out a bias here in not having a big enough sample size. A larger sample size will usually make the data better, so it is possible sampling a bunch more teeth may result in different values. Still though, the researchers figured that the low zinc values in both samples from extant great whites compared to other living sharks is in line with the generally high trophic level estimates of the species. The absence of significant zinc value differences within many species among the different locations and times suggests there is only negligible differences in zinc value food web baselines. Therefore, the team figured that the variability in zinc values over space and time for Otodus and Carcharodon reflect true dietary differences, which could mean quite a lot for the ecology and evolution of both lineages on a local, global, and time scale. The oldest species of the two shark lineages are Otodus chubutensis and Carcharodon haestalis from the early Miocene. Carcharodon haestalis had higher zinc values, meaning it was on a lower trophic level than the larger big-toothed shark. That makes perfect sense, as Otodus chubutensis may have reached as large as 12.2 meters, 40 feet in length. Oddly enough, the zinc values for Otodus chubutensis were the lowest in the entire dataset for this research, even lower than Megalodon, meaning it could have occupied a higher trophic level than both Megalodon and the contemporary great white species Carcharodon haestalis. An important check and balance to this comparison to the contemporary Miocene megatooth and great white sharks is that the lowest mean zinc values recorded in the dataset do not appear to be at all related to a shift in diet as the animals aged, as in a different ratio between adults and juveniles. In fact, the team found no correlation between zinc values and total body length, as the teeth they sampled were from more mature individuals, so if there were dietary shifts as they aged, they would have already shifted before they shed their teeth. When only including the Otodus specimens from the Atlantic and Paratethys/Tethys regions, the team found significant differences in values between Otodus chubutensis and Megalodon. During early Pliocene times, Megalodon shows an increase in mean zinc values for the Atlantic populations, which hints at a reduction in trophic position. At the same time, the contemporary great white species kept the same zinc values as they did back in the Miocene with the haestalis species. What's weird here now is that modern great whites seem to have a lower value, and thus a higher trophic level than both the Miocene and Pliocene species and Megalodon. Living great whites prey primarily on high trophic level enjoying marine mammals, though the ancient great whites and megatooth sharks were likely opportunistic in what they ate, like pretty much every predator ever. Fossil evidence in the form of bite marks left in bones suggests that both ancient great whites and megatooths fed largely on marine mammals like whales and pinnipeds, just like the modern great whites. It remains unclear if these ancient feeding traces were from active predation or scavenging, and how important each prey species were to the diet of each shark taxon. Early Pliocene great white and megatooth zinc values tell us that lower trophic level mammal prey, like baleen whales and sirenians, or manatees and relatives, may have been important for both of them. Baleen whales, being filter feeders, have higher zinc values in their tissues than the toothed whales or pinnipeds. Bite marks on late Miocene to early Pliocene baleen whale bones from great whites and megatooths corroborate, at the very least, occasional feeding events. Extinct small to medium-sized baleen whales, members of Mysticeti, were common and abundant during the early Pliocene, making them easily available prey items for large sharks. In contrast, early Miocene-aged whale fossils are dominated by toothed whales. The early Miocene European and North American sites sampled in this study did not have any baleen whale fossils. The lower zinc values and higher trophic level of early Miocene otodus and modern great whites may be partially related to the lack of lower trophic level mammals to eat. Baleen whales, the higher trophic level mammals, became far more abundant following a plateau in diversity during the mid-Miocene, which may coincide with the mid-Miocene climate optimum or mid-Miocene disruption, which saw a wave of extinctions of terrestrial and aquatic life from a relatively steady period of cooling, resulting in a growth of global ice sheet volumes and re-establishment of the ice of the East Antarctic ice sheet. Therefore, baleen whale remains become more abundant in late Miocene to early Pliocene fossil assemblages from North Carolina and Florida, which were sampled, measured, and analyzed in this study. Where megatooth and great white fossils were found together in early Pliocene sites from North Carolina, the zinc values overlapped, meaning they were in the same trophic level. Since biology and ecology is far more complicated than a simple food web, two species overlapping in trophic level doesn't inherently mean they were in direct competition with one another, since they both could have been eating different prey that just happened to be in the same trophic level. Despite this caveat, some overlap in food items between the species is still pretty likely, as indicated by fossil bite marks. Living predatory sharks tend to eat just about anything that moves and that they can kill, and there is some evidence of modern great whites eating everything and not just the marine mammals they are adapted for, with even some evidence of some great whites specializing on being at a lower trophic level. Higher levels of individuality in diet and opportunism are possible explanations for the range in zinc values recorded in great whites and megatooths from Pliocene North America. The extinction of Megalodon likely occurred due to multiple reasons: climate change, thermal limitations, collapse of prey populations, and resource competition with great whites and toothed whales. The zinc values collected and compared and contrasted in this study suggest there was a major trophic change, with megatooth sharks decreasing in trophic level from Chubutensis to Megalodon, and great whites increasing in trophic level from Pliocene Carcharias to modern Carcharias. If the values found in this study hold up in the future, it may imply strong competition between the lineages for prey items. In other words, the great whites played a hand in the extinction of their bigger cousins. For more interesting stories about nature, the history of life, or what goes bump in the night, subscribe, hit the bell icon for updates, like this video, and drop a comment in the comment section below. Thanks for watching. Special thanks to my elephant tier patrons: Arda Bayer, Biotiverse, Christoph Hubinger, Dinosaur, Isaiah Garza, P.A. Brew News, Ray, Rudy Redgrave, Smiling Walrus. And another thanks to my Tyrannosaurus tier patrons: Iberospinus, Iron Bladesman, Swaffles is Weird, Teeny Dragator, The Dogman,